Okay, so my senior thesis and research is entitled Effects of pH20 hyaluronidase Inhibition in Vivo Following Cerebral White Matter Injury. I know that's a mouthful, but my name is Rachel Siltman, and I had the pleasure of working in Dr. Stephen Back's lab, departments of pediatrics and neurology, as well as Dr. Tashin Srivastava. So basically, the importance behind this research is that 15 million babies are born preterm each year, and in the United States alone, 10% of all live births are preterm as well. And prematurity is the leading cause of abnormal brain maturation that leads to a spectrum of neurological and motor defects in survivors of preterm birth, including cerebral palsy in severe cases. So white matter injury, what is white matter? White matter is kind of the interior part of your brain, and it's composed of myelin, which basically encompasses the axons along a neuron that I have pictured here. And so infants born between 23 and 32 weeks have a predisposition towards this injury. And this is kind of due to the fact that preterm infants have a narrow autoregulation window, which basically influences the cerebral blood flow, which is the blood flow going to the brain decreases as the arterial blood pressure decreases as well. And that's basically the pressure exerted by the aorta on your arteries. And so this leads to inefficient or reduced oxygen and blood to the brain and lungs, and this injury is termed hypoxia ischemia. So the injury response, what happens? Basically, this picture is a schematic diagram of our lab that has defined a novel mechanism of this myelination failure. So basically when the myelin is not generated, and as you can see, the red line that I have pictured here indicates where the maturation stops. And there's basically when this injury happens, there's an acute cell death. And this is kind of followed by a two-phase injury response, a vigorous proliferation being the first phase, and then the second phase being that these newly generated cells fail to mature, as indicated by the red line. So extracellular matrix breakdown. First of all, the extracellular matrix is basically the components that are secreted by the cell that help in functional and structural support of the brain. But the molecular mechanisms of this breakdown are poorly defined. But we do know that the blood-brain barrier, or basically the barrier that separates your brain from the rest of your body, is compromised. And this leads to a leaky brain, meaning that molecules and components can come in and out of the brain that usually should not. And this results in a glial scar that forms. And that's basically due to the acute cell death and then myelin along the axons not being generated. And the hyaluronic acid is a major brain component as well, and altered levels are found in this glial scar that are not beneficial. And so this HA, or hyaluronic acid, is digested into these toxic bioactive fragments by an enzyme called pH20 hyaluronidase. So for this experiment, we investigated white matter injury by using a new drug called SCBR3 that's never been tested before, to block the generation of these toxic fragments by the enzyme hyaluronidase. And so our hypothesis was that the SCBR3 mediated inhibition will attenuate the injury response that we observed during white matter injury. So our methods for this, we used an animal model, so a neonatal rodent preterm equivalent model to a fetus, a human fetus in the womb. And at time of surgery, these animals received a single injection of this drug or served as controls. So after the animals were euthanized, these brains were fixed and stained with antibodies. And here I just kind of have a picture to show you guys what this looks like under the microscope. These are 3 antibodies, the red, green, and blue, and this is kind of all of them superimposed on each other, so this is what it would look like. So how we quantified and our statistical analysis. So we used a microscope and a fancy software called Stereo Investigator, and this basically allowed us to look at tissues under the scope, 3D tissues under the scope. And the probe called the optical fractionator basically allowed us to go through the tissue and count all of the cells that we marked with the antibodies. And so this is this is a slice pictured under a scope of a rat brain, and the blue and red box indicate our area of interest. And basically this kind of like bird-shaped thing is called the superior corpus callosum, and this is rich in myelin. So this was our area of interest. And the white arrow indicates where we injected the drug. So this is a control and an injured or hypoxic ischemic slice of a brain. And as you can see, the injured slice has a lot of red in it. And the red is basically an astrocyte marker, which is really important in the injury response. So a lot of astrocytes indicates that there is injury. And as you can see, there is a tear down here. So this is kind of just comparing the two. This diagram is kind of the crux of our experiment. And this sums up basically our findings as well. The SCBR3 pictured here is our experimental group. And this is basically the animals that underwent surgery and were injected with the drug. And as you can see, it significantly decreased compared to all other controls. And this was indicating that the drug was doing something. It was blunting this proliferative response. So basically wrapping up the research from all of our findings, we found that the hyaluronic acid bioactive fragments drive an injury response in vivo, and that this drug called SCBR3 was potently blocking that cell proliferative response in white matter lesions generated by hypoxia ischemia. So our future studies would use more animals, a bigger cohort to experiment with, as well as characterizing and examining all of the cell types that could be affected by white matter injury. These are all the people I would like to thank for helping with my research and making this happen. These are my references, and any questions.
Well, Rachel, you did an amazing job of condensing it so that a non-biology person could kind of understand what you're doing with that thing. So thank you very much for that. So how is this leading on to other work in your life?
So the plan so far is to apply to PA school, but I love research, and this kind of solidified my desire to be in research still. So I would like the best of both worlds, to do research, but then also do the clinical side as well. So yeah, I made great connections at OHSU and Dr. Back's lab, so I hope to continue that. Yeah.
Is Dr. Back's lab, it sounds like they're going to continue their work with this particular drug to see if it could be something that improves what happens in white matter injury.
Correct, yeah, they're actually, he's finishing up a grant to get more money to pursue this. This is kind of another rabbit hole, as you say, that they're gonna explore. And so that's really exciting. They're waiting for approval on that. That's it? Oh, yeah.
So your studies were with your rat, you were using a rat model?
Yeah, rat model.
So you were kind of mimicking what might happen in a preterm baby, or what could happen in their brain?
Correct, yeah. So I didn't get super into the methods because it gets confusing, but basically a P3 or a postnatal day, 3 days after a rat is born, is the equivalent to a 5-month-old baby within the womb in terms of brain. So their neurons and oligodendrocytes and that sort of thing. So yeah, this is a defined model that has been used for years. So that's what we used. Thank you.